Hello everyone. Today I am going to present about culture method of polychaetes. But before that, I am introduce myself. My name is Nurul Ashikin binti Ismail and I will be present on behalf of my team. So this is my team members, Sofia, Sabrina, and Hashima. Okay, let's proceed to the culture of polychaetes. So this is the first species that we identify and we search through the journal, which is culturing larva stage of Polydora cornuta. So the simplest method of rearing pelagic marine larvae involves close culture of this Polydora cornuta, which in the culture medium is not continuously changed as in the flowing seawater system. Closed culture are the only practical alternative for inland laboratories without access to natural seawater. Successfully closed culture system only require a means of moving the culture water to keep the larvae suspended and periodically changing the water to reduce the concentration of waste product. Okay, next. We continue as a culture water that we use in culture Polydora cornuta was used artificial seawater, or the terms of short is ASW, mixed in 50-gallon batches using tap water and aerated constantly. Several clean clam shells, which are the species known Marcenaria species, were added to the tank because of reports that unknown factors in the shells was improved marine animal culture in the laboratory. So the filter ASW through a 5 micrometer bottle top filter was used, and failure to filter the water will be contaminate the crustacean and algae into the culture. So 4 liters of artificial seawater were used in each vessel. So let's continue. Still we in the same topic, which is culturing Polydora cornuta. Okay, one of the half water was changed twice weekly by pouring the water into the sieve shown in the figure A that was shown at the left screen, which was filled with water above the level of mesh so that the larvae are not forced against the screen or allowed to dry out. The mesh size should be based on the size of the larvae to be maintained, which use 50 micrometer for young larvae and 100 micrometer later in the development. Once the water has run through the sieve, the concentrated larvae are gently poured back into the culture vessel and the sieve in backwash with a squeeze bottle of artificial seawater to release any larvae caught on it. The culture were maintained on a normal light or dark cycle, but it should be noted that based on the other researchers, Dean and Mazukris, 1975, have reported faster growth of Polydora cornuta was kept in constant darkness. Okay, next. This is another species of polychaetes that we have been identified or we search, which is Arenicola marina. So as you can see at the left, at the right, at the screen, this is the book that we search. The name is Advanced in Marine and Brackish Water Aquaculture. So the summary we got from this book is: first one, collection of broodstock, breeding and development. So other worms are collected from the habitat. The worms can be kept in room temperature, aerated aquaria in 32 ppt artificial seawater, and adults are sexed according to the color of their gametogenetic parapodia. Oxide are obtained by cutting off a single parapodium from a gravid female and emptying the contents, which is several thousand eggs, into a 150 micrometer screen immersed in 0.2 micrometer filtered artificial seawater, known as FASW, at 32 ppt salinity in a 35 millimeter petri dish. Then they will be transferred into 100 millimeter petri dish of fresh water, filter artificial seawater, and then incubate them for about 40 minutes before fertilization occur. Then the dry sperm is extracted from the optimal amount of sperm suspension to be used to obtain complete fertilization without excessive polyspermy. Swimming ciliated bastulae developed by 12 hours at room temperature. Okay, the second one is simulated beach. So this one is developed for harboring adult worms for culture. So it made by depositing about 3 centimeters of sediment from the collection site into a wide shallow vessel deep enough to allow for 6 to 8 centimeters of water on top. The last one, food for culture organisms. Each individual can feed once in every 3 days with commonly available commercial food used for tropical fish. Several types of algae can be used, but Dunaliella salina, a green algae, and Isochrysis galbana, a golden brown algae, give adequate nutrition and they are easy to maintain in culture. Okay, next. The last journal that we search for the culture method of polychaetes is another species is Perinarius cf. Nuncia. So we summarize from this journal the firstly, male and female sexually matured adults, epitocs of PCF nuncia were selected and artificially made in a polystyrene box in a clean seawater. The male to female sex ratio was 1:2. Then the epitocs were cut into pieces to release free sperm and eggs from the coelomic cavity of males and females, respectively, which were gently mixed in seawater for 3 to 5 minutes. Next, the fertilized oocytes were transferred into a 500-liter plastic tank containing clean seawater with adequate aeration. And lastly, the larvae reached the stage of nectochaeta, 3 centigerous segment within 48 hours. So that's all for the presentation from my group. Thank you.